We ready? Good morning, everyone. I've been asked to tell you why I endowed this annual lecture, and the answer is quite simple. I want to memorialize my husband, Bob Adler, who was an incredible father, grandfather, husband, and lawyer, and who had the need for and received the most incredible medical care from Dr. Louis Amorosa. I also want to honor Dr. Amorosa, whose almost 39 years of medical care for Bob facilitated Bob being able to live his best life. And I want him to be able to select an annual lecturer. This inaugural year, I'm grateful he chose Dr. Kahn, who actually, parenthetically, I was privileged to meet a couple of weeks ago, sadly, at my cousin Jesse Roth's funeral. Jesse was also a leading endocrinologist, and I believe worked with Dr. Kahn. Welcome, Dr. Kahn. And thank you, Dr. Amorosa, for caring for Bob and for all that you do. And I'd like to introduce Dr. Buettner, sorry.
Good morning, everybody. My name is Christoph Buettner. I'm the Chief of Endocrine, and it's my distinct pleasure to introduce Dr. Ronald Kahn, who is currently the Mary Iacocca Professor of Medicine at Harvard Medical School and the Joslin Diabetes. Dr. Kahn has been tremendously influential in the field of diabetes, is among the most published physicians throughout science. He really has defined and elucidated the role and function of the insulin receptor, of molecules that participate in insulin signaling, such as IRS. He also has done a lot of work in adipose tissue biology and defined the role of proteins secreted by the adipose tissue that then impact metabolism. So among his thousands of publications and the many awards, he is a member of the Academy of Medicine, a member of the Academy of Science. He also reminded me at dinner that funny story that in the end of in the 90s, he founded a company with his friends Jeff Lyer and Bruce Spiegelman, and one of the goals was they had bought the rights to GLP-1 that Joel Habener, also at Harvard, had cloned, and one of the goals was to develop GLP-1s into therapeutics for diabetes at a time when nobody really believed that that was a very viable option, given that it primarily at the time caused nausea. All of this was supported by Pfizer, and after a couple of years, even though they had made progress, unfortunately, Pfizer pulled the plug, and the rest is history. Dr. Kahn also has a tremendous legacy in the many fellows that he trained, more than 200. Indeed, the very first time I met Dr. Kahn was at a spontaneous engagement party in Boston, where I met a couple of fellows from his lab, and I quickly realized that this environment was very, very outstanding to get trained as an endocrinologist and a diabetologist. And there was a steady flow of international fellows from literally all continents going back and forth to Harvard. Some stayed in the US, others went back to their countries, and now are leaders in diabetes throughout the world. So where did Dr. Kahn receive the knowledge to be such an outstanding mentor? And that's where Jesse Roth comes in, who happens to be your cousin, Toby and Larry. And Jesse Roth was still a young physician when he started to mentor Dr. Kahn at the NIH. He mentored Bob Lefkowitz, who later became a Nobel Prize winner for elucidating G-coupled protein receptors. He also mentored Jeff Lyer, who later became the dean of Harvard Medical School, and many others. Eventually, he actually settled at Northwell and lived in Queens. And that's where I came in contact with him because when I was a junior faculty at Sinai, he seemed to attend every diabetes meeting possible in the city, whether it was Sinai or Einstein or Columbia. And he was a very well-respected giant, physically also very tall. Everybody knew him, and he knew everybody. Had a lovely smile and a lovely laugh, and was a real mensch. So with that, I want to ask Dr. Kahn to come to the podium, and we have a plaque for you. So this is for the Robert Adler Memorial Lecture, which is the inaugural lecture to Ron Kahn. Yeah. Right. All right. With that, the stage is yours.
All right. Well, thank you very much, Christoph. It's really an honor to be here, a pleasure to be here. I had a great opportunity to meet with some of the faculty last night and to give this first Robert Adler lecture. And since I guess nobody else showed a picture, let me show you a picture. Robert Adler. I did not know Mr. Adler, but I certainly got to meet Toby a little bit over another occasion and heard about what a great individual he was, a sort of a champion for civil rights through his law practice, and that he really did a lot, I think, of good in his law practice. He also had this medical connection, as you heard through Toby, who was, in fact, the first cousin of my mentor, Dr. Jesse Roth, whose name has been mentioned a few times, and who passed away literally just about a month ago. And he was another giant of training and who really gave me an appreciation not only of science, but of humanism. So I must run in the family that there's this large thing and all the people they touched, Dr. Omarosa and so many others. So with that, I'd like to show you my disclosures. Nothing I present today is really influenced by any pharmaceutical companies, although I do consult from several new startups. And my learning objectives are to teach you a little bit about the growing epidemic of obesity, type 2 diabetes, and metabolic syndrome at the population level, to talk to you a little bit about insulin action, because that's what I work on, and the role of insulin resistance in these disorders, and how we're finally, I think, at a place to begin to develop tools to get to the molecular basis of this insulin resistance, which is still the major part of type 2 diabetes for which there is no direct treatment. So the major pathophysiologic driver of type 2 diabetes, as I will show you, is in fact insulin resistance. And short of exercise and diet, none of the drugs we have directly address that problem. I will tell you a little bit new about adipose biology, and then because I'm at the home of Marty Blazer, I don't know if he's in the audience today, I have to say something about the microbiome. So will do that. So just a little population genetics, and I think most of the fellows and residents and house staff are quite aware that diabetes and obesity are really parts of worldwide epidemics. In the US, there are currently over 50 million people with diabetes. 95+% of those are type 2 diabetes, and that number is expected to grow by 21%. And in most of the more westernized countries of Europe, Australia, there are similar prevalences and similar growth rates. But the real problems for diabetes worldwide are actually in other countries. So in South America, in Southeast Asia, where historically the rates of type 2 diabetes have been lower, the growth rates are much faster, 45 to 73% predicted over the next 25 years. And it's even worse yet for Africa and the Middle East, where up to 100 or greater than 100% will be an increasing prevalence of diabetes in these countries. So that overall, in the next 25 years, despite everything that's going on, the WHO still predicts almost a 45% increase in diabetes worldwide. In the United States, we have actually a kind of a geographical differential as well. Up here in the mid-Atlantic states, you are lumped with the center of America, which has the highest prevalence of diabetes. We in New England have a lower, excuse me, a lower prevalence, and the rest of the United States is somewhere in between. But what I wanted to show you this map for is to point out that type 2 diabetes is really a kind of a canary in the coal mine for other metabolic diseases. And so there are parallel epidemics of obesity and metabolic syndrome, particularly metabolic associated fatty liver disease and cardiovascular disease. And so wherever you are in the world or in the United States, this is going to be a growing problem. Now, a lot of effort has been made to understand the genetic drivers of type 2 diabetes. And actually, I'm going to show you a surprisingly old slide. This slide is from 2012, so more than 12 years or 13 years ago. This is the first 100 type 2 diabetes genes associated. And you can see starting back in the year 2000, using genome-wide screening, various investigators looked for what are the genetic drivers of this very common problem. And many genes have been identified. As you can see on this is the first 100. But I would say that as of 2024, last year or so, there were over 800 SNP polymorphisms associated with type 2 diabetes. Over 90% of these are in non-coding variants. They don't change the structure of the gene. And I would say that overall, most are still not functionally defined in how they contribute to diabetes. So even though genetics has opened up a lot of new information, I would say, as a non-geneticist in a way, it's been a little bit disappointing that it really hasn't given us the smoking gun to look for the therapeutic target to change this epidemic. So one of the things that's going on a lot now is to try to reclassify diabetes and see if we could get maybe better insight this way. There have been several articles over the last 5 years saying that maybe we need to reclassify and get away from type 1 and type 2 to maybe a more functional classification. And indeed, type 1 diabetes is now sort of renamed in some spheres as severe autoimmune diabetes. This includes classical type 1 diabetes that we see mostly in childhood and young adulthood, although at any age it can occur, but also something called LADA, or latent autoimmune diabetes in adults, because type 1 diabetes can occur at any age. And of course, these are individuals with autoimmunity, producing antibodies to GAD and to insulin itself, loss of beta cell function. But type 2 diabetes is also now being subdivided clinically by some individuals into those with more severe insulin deficient forms of type 2 diabetes, those with more severe insulin resistant, those that are more obesity associated, those that are more age associated. And you have this kind of proposed new classification. I don't think anybody clinically is really using this yet, because it really still hasn't gotten us to saying what are the drivers. And I think that this is one of the things that we need to think about as physicians and physician scientists, is classifications are fine, but ultimately we want a classification that will get us to the root causes and then maybe to new and different ways to treat these subgroups, and we're still not quite there. What I would like to make a point of is that all of, I'll call it garden variety type 2 diabetes, is really driven by insulin resistance. So starting back, excuse me, just got a little cough here. Starting back about 1975, an investigator at the Joslin named Stuart Solner decided, how are we going to figure out what's the earliest defect in type 2 diabetes? And because the Joslin had a lot of patients with type 2 diabetes, he got this idea. Why don't we take couples where both the husband and the wife have type 2 diabetes, and we'll look at their children, because they've obviously are very enriched for whatever genes drive type 2 diabetes. And we'll do a bunch of metabolic testing while they're normal, and then we'll follow them and see who gets diabetes or not. And he measured things like insulin secretion, and you can measure acute insulin secretion first phase or delayed insulin secretion second phase. He measured insulin sensitivity using a frequently sampled IV glucose tolerance test. And he measured something called glucose effectiveness, that is, how much glucose can get in the cell even if insulin levels don't go up. And then he asked who in these different characteristics will develop type 2 diabetes over a 25-year follow-up. And the results were remarkable. First of all, first and second phase insulin secretion, sorry, Christoph, did not predict who was going to become diabetic. Insulin secretion eventually becomes disordered, but at this early phase, there was no abnormality that he could find in insulin secretion. But insulin sensitivity, or low insulin sensitivity, insulin resistance, and low glucose effectiveness, low ability of glucose to get in the cells, were predictive. So if you divided this offspring population into 4 groups, those individuals with low insulin sensitivity and low glucose effectiveness, just in the lower half, and that's shown here by the red curve, over 80% of those individuals over a 25-year follow-up would become type 2 diabetic patients. Whereas if you took individuals with high insulin sensitivity and high glucose effectiveness, literally none of them became type 2 diabetic, even though they had 2 type 2 diabetic parents. So this differentiation between insulin sensitivity and glucose uptake and metabolism really distinguished at the extremes the most likely to develop diabetes and the least likely to develop diabetes. And if you had one low value and one high value, then you were at some intermediate risk group. So insulin sensitivity is there, insulin resistance is there, and ultimately what we need to understand is both what drives the insulin resistance and what leads it to ultimate hyperglycemia and type 2 diabetes. And of course, eventually the beta cell does lose its ability to compensate. I always say it's like the weightlifter trying to lift this heavy burden, and eventually they can't lift the heavy burden, and so it does fail. And that's another important component that will trip people from maybe insulin resistance to true hyperglycemia. And we have our usual players in this game: genes, genetics, and epigenetics, because epigenetics could be a bit important drivers, and of course, environment aspects, including our diet and the effects of the diet on the microbiome. And so what I'm going to try to do today is explore a little bit about what these tell us now about the pathophysiology of type 2 diabetes. At the same time, I want you to keep in mind that type 2 diabetes, in one sense, is only one part of what these factors are driving. Insulin resistance is not only central to the development of type 2 diabetes, it's central to the development of dyslipidemia. It's of course important in central obesity and hypertension, accelerated atherosclerosis, the classic metabolic syndrome, as well as now this ever-expanding metabolic syndrome, which includes metabolic-associated fatty liver disease, MAFLD and MASH, reproductive dysfunction, like polycystic ovarian disease, is associated with insulin resistance. Patients with insulin resistance have increased risks of a number of cancers. They have definitely increased risks of Alzheimer's disease and ultimately impaired longevity. So when we look at these driving factors, genes, epigenetics, environment, microbiome, we want to look at them, how they drive insulin resistance, not just to diabetes, but also could drive to these various other manifestations of the insulin-resistant or metabolic syndrome. So to do this, I'm going to teach you a little bit about what is the current state of understanding insulin action. Where are we now in 2026? We're now 105 years since the discovery of insulin, the molecule that's on my tie. We now know a lot about how it works. When it was first discovered, of course, nobody really knew how it worked, but now we know. And this is a schematic of the insulin receptor. It's a tetrameric protein. It's composed of 2 alpha subunits and 2 beta subunits that are linked together by disulfide bonds. And the alpha subunits are completely in the extracellular domain, that's the ECD. The beta subunits have a part in the extracellular domain and then have a transmembrane domain and an intracellular domain. And this intracellular domain contains a site for binding ATP and a catalytic tyrosine kinase activity. It's a tyrosine kinase receptor, like many receptors for growth factors and some other hormones. And we can now actually visualize, using scanning EM, we can visualize the insulin receptor protein, as shown here in these diagrams, where you can actually see the alpha subunits out here, the beta subunits here, and this is the stalk of the transmembrane part. This is the extracellular part of the insulin receptor. And this has allowed people to actually make three-dimensional crystal structures. So one of the ways that pharmaceutical companies look for ways to activate receptors and molecules that are enzymes actually takes advantage of our ability to structurally model the receptors. So this is mostly the alpha subunits of the piece of the beta subunit here. Shown up here in this sort of reddish and bluish color is the insulin molecule itself bound to the receptor. And the tyrosine kinase is on the other side of this transmembrane domain, waiting to be activated by receptor binding. So the insulin receptor sits here in the membrane of the cell, and there's a very closely related receptor called the IGF-1, insulin-like growth factor 1 receptor. Very similar structure, kind of similar molecules, and both of these sit in the membrane of the cell. And when they bind their respective ligands, insulin in one hand and IGF in the other, and there's some cross-reactivity, but at a low level of these two ligands for the other receptor, then these tyrosine kinases are activated. They stimulate their own phosphorylation, and they stimulate the phosphorylation of a series of intracellular substrate proteins we call IRS proteins, insulin receptor substrate 1, 2, 3, 4. Very clever nomenclature here, IRS 1, 2, 3, 4. And each of these is phosphorylated on many sites. And these actually serve like an intracellular messenger, just the way cyclic AMP serves as an intracellular messenger for some receptors. These proteins serve as intracellular messengers, and they do that by docking or binding to other proteins inside the cell and activating them. And the most important pathway that's activated by these receptors for metabolic actions is called the PI3 kinase pathway. That stands for phosphatidylinositol-3 kinase, which is itself an enzyme composed of a regulatory subunit and a catalytic subunit that, when activated, generates a lipid mediator, phosphatidylinositol trisphosphate, or PIP3. And this PIP3 then binds and activates other downstream kinases. These are serine and threonine kinases like AKT, protein kinase C, mTOR, and so forth. So you can see from this cascade, this network, that ultimately this signal that comes out of the single insulin receptor can create a divergent pattern of activities. Some of these activities are important to get glucose uptake. And how does that occur? Insulin-sensitive glucose transporters in muscle and fat. These are the GLUT4 glucose transporters. They're inside the cell in vesicles, and these vesicles translocate to the membrane and allow glucose to come into the cell. So we stimulate glucose uptake. Other parts of this pathway stimulate enzymes involved in lipid synthesis, in protein synthesis, in glycogen synthesis, and ultimately in the liver, turning off gluconeogenesis, because when insulin levels are high, you don't want to make more glucose. You want to turn off glucose production and get it metabolized. There's also a second pathway that comes out of these two receptors, more so for IGF-1 than for insulin, and that involves a different substrate called SHIC, which activates the RAS-MAP kinase pathway. And I bring this up because this is a growth pathway. So insulin is both a metabolic hormone and a growth hormone, as is IGF-1, a growth hormone and a metabolic hormone. But the preference of insulin is for the metabolic side, and the preference of IGF is more for the growth side. This is actually clinically important also to keep in mind in a couple of situations. For example, in certain situations where insulin levels are high, insulin can spill over and activate the IGF-1 pathway. It does this in fetal macrosomia. So infants of poorly controlled diabetic mothers develop hyperinsulinemia, and it promotes fetal overgrowth. Part of the problem of obesity is probably linked to some level of growth stimulation for adipose tissue by these high insulin levels. And for those of us who see patients with severe insulin resistance, we know that severe insulin resistance is marked by a skin overgrowth condition called acanthosis nigricans that appears like a dark velvety part of skin. And this is really stimulation by the high insulin levels through IGF-1 receptors of a growth pathway. Conversely, there are spillover in the other direction. So some non-islet cell tumors, sarcomas and other types, can make insulin-like growth factor 1 related molecules. And those bind to the insulin receptor and create tumor hypoglycemia, even though insulin levels are low. So this idea of spillover of the activity of one hormone on the other receptor, what's something that Jesse used to call specificity spillover, is a real phenomenon, and it's clinically important to be aware of as part of the pathophysiology of these diseases. So where is the defect in insulin resistance? I showed you this elegant pathway, and I can tell you that we've studied this for quite a while. We studied all these steps, and many of them are abnormal, but where does it start? And how could we begin to get to the core of this? So one tool I want you to be thinking about as you think about not just this disease, but other diseases, is now the ability that we have to make cellular models that are human models, not just animal models. And for example, we can take iPS cells, induced pluripotent stem cells. We can make them, as I'll show you, from blood cells or other cells, and we can make them into liver, muscle, fat, brain type things. So we can now study cells from an individual with type 2 diabetes or another disease in vitro and really try to understand where this all starts. And this is what a system that we think is very important for future studies as well as current studies. So we've taken cells from type 2 diabetic or insulin resistant donors. You can genetically reprogram them to iPS cells, these induced pluripotent stem cells, in vitro, and then you can grow these cells essentially forever. So you've got some sample of that individual in vitro forever, and you can make them into all different kinds of cells, beta cells, muscle cells, liver cells, and so forth. And we've done this using different kinds of initial material. You can do it from a skin biopsy, peripheral blood cells, a muscle biopsy. You can use a lot of different ways to reprogram them, and you can make them into muscle, liver, fat, beta cells, or any cells. So this allows us, for the first time, to get in a laboratory a humanized model of insulin resistance. And we can do this across the spectrum of insulin resistance. We can do it with individuals who have mutations in the insulin receptor, individuals with type 2 diabetes, or individuals who have insulin resistance but don't yet have type 2 diabetes, to ask, how do their liver or muscle respond in terms of insulin signaling, glucose uptake and metabolism, RNA and protein expression, and signaling measured through a more sophisticated approach called phosphoproteomics. I just want to show you a little of this. I know that this is maybe a little deeper than you want to go, and I'm trying to show you this not so much that you learn all of the things, but you learn that we can now model this in a way that we couldn't before. And this opens tremendous opportunity to find the primary defect and to develop new therapies. So let's take type 2 diabetes as our starting point. So a number of years ago now, we took a population of 8 normal middle-aged individuals and 8 type 2 diabetic individuals. They were about 60 years of age. They were sort of half male and half female. On average, of course, the type 2 diabetes patients had obesity compared to the controls. They had higher glucoses and higher hemoglobin A1Cs. And from these individuals, we made iPS cells. And then we could make the iPS cells into skeletal muscle cells and say, what is the nature of insulin resistance outside the body for these cells? Are these cells already insulin resistant? So here's that insulin signaling cascade, the insulin receptor, the IRS proteins, PI3 kinase, AKT, glucose uptake, and so forth. And when we did this and we measured the activity, let's say of AKT, a critical enzyme for all the metabolic actions of insulin. You can see in the control cells, insulin stimulates about fourfold AKT activity. And in the cells from the type 2 diabetic patient, in this in vitro situation, they still stimulate, but instead of stimulating fourfold, they only stimulate threefold. So there's insulin resistance. It's quantitative, not absolute. You can overcome it if you give enough insulin, but there's a defect in signaling. And this defect in signaling at AKT is also seen further downstream at the enzyme called glycogen synthase kinase 3 and at this transcription factor called FOXO1. And because it's there, these cells, compared to control cells, have decreased ability to take up glucose when you stimulate them with insulin. So the iMyos, the iPS-derived myoblasts from people with type 2 diabetes already are insulin resistant in vitro in the absence of all these circulating factors that might contribute to insulin resistance. And we can go further. We can do all these omics. We can do RNA analysis, DNA analysis, metabolomics, phosphoproteomics. And I just show you this to show you the power now of the tools we have. And by doing phosphoproteomics, we measure thousands, literally thousands of sites that are phosphorylated in response to insulin. And this is a heat map showing you 1,172 of these sites. The top sites are insulin action sites. You see basal phosphorylation is low, that's the blue color, and insulin stimulates it, that's the red color. And it does it in controls and type 2 diabetics and in males and females. And here are some sites that insulin decreases. But what I want to show you more is that in vitro, there are many sites which are not insulin regulated, but already have abnormally high phosphorylation in type 2 diabetes, or have abnormally low phosphorylation in type 2 diabetes. So there are literally hundreds or perhaps thousands of sites that are altered in their phosphorylation by either insulin or insulin resistance. And in some cases, the differences are even sex-specific. So here are sites which are different between the males and females, something we never thought about. Is there sexual dimorphism in the pathophysiology of diabetes and other diseases? And I can say at the cellular level, cells from males and cells from females do not show the same gene expression, nor do they show the same phosphoproteomics. And it's not just the X and Y chromosome that makes the difference, it's things all through the autosomes. And we can do this in the type 2 diabetics, and we can do it in the insulin-resistant non-diabetics, so we can try to say, what is the core of these defects? What are these key altered roles? And I'm not going to try to expect you to memorize this in any way or even know it, but what I think you should really know is that in type 2 diabetes, so far when we use this tool, it doesn't point to just one spot of abnormality. It points to many abnormalities, and trying to figure out which is the key one is really a challenge. For example, in the insulin receptor substrates, there are sites that are overactive or upregulated in both diabetes and non-diabetes. There are sites that are downregulated. And throughout this whole signaling cascade, through the cytoplasm and even into the nucleus, there are literally hundreds of sites that are changed. So our challenge is to try to use this kind of material to get a new insight to the pathophysiology of type 2 diabetes, and then to find new ways to treat it. And this is true not only in muscle, this is true if we make them into liver cells. We see similar kind of signaling defects. And these liver cells show exactly the defects of type 2 diabetes. That is, insulin is supposed to turn off glucose production. This is the enzyme responsible for glucose production, and insulin normally turns it off. And in type 2 diabetes, there's more activity of this enzyme, and it doesn't turn it off. You can't turn off hepatic glucose production as well in an insulin-resistant diabetic cell. What's interesting in liver is that insulin also stimulates lipogenesis, production of lipids and lipoproteins, and that is insulin stimulated both in the control and even hyperactive in the type 2 diabetics. So this is what we call pathway selective insulin resistance. One is reduced and one is overactivated. This helps us understand from this kind of molecular study, what is this link between type 2 diabetes and metabolic-associated fatty liver disease, or what used to be called non-alcoholic fatty liver disease, because diabetes promotes the risk of steatohepatitis and cirrhosis and hepatocellular carcinoma, and NAFLD makes insulin resistance worse and promotes atherogenesis. And all of this comes back to that circle with the rays of arrows coming out of it that shows you that insulin resistance can drive diabetes, it can drive fatty liver disease, and if you made a similar diagram for ovarian function and other cancer growth, you would see there's always this crosstalk between the two. Now, you can't talk about type 2 diabetes without talking about the most closely associated other common physiologic problem, and that's obesity. And obesity has literally been a problem for 20,000 years. In fact, I was first shown this slide, or a variation of this slide, by my mentor Jesse Roth probably 50 years ago, right? Because he was also interested in art, and he recognized going to the museum that if you look at these ancient Venus statues, the Venus of Willendorf, which resides in Vienna now, or the Venus of Lespuge, that resides in Paris, that the Venus on the left has what we call central obesity with a high risk for insulin resistance and metabolic syndrome. And the one on the right has more peripheral or buttock-associated obesity and probably is at lower risk for insulin resistance and metabolic syndrome, apple-shaped and pear-shaped. And I would say that on top of the genetics of diabetes, obesity is a really, really, really important confounder. So this is a study published by Jose Flores about, I guess, 7 or 8 years ago now, where he took a population that was genotyped and followed for the development of diabetes over 10 years. And I'm showing you here these red lines at the bottom of this graph represent a genetic risk score in the lowest quartile of risk to the highest quartile of risk. And you can see that as your genetic risk score goes from very low to very high, during this 10-year follow-up, your likelihood of developing type 2 diabetes is, in fact, increased substantially, maybe 3 or 4-fold. So genetic risk does predict who's at higher risk. But if you add on top of the genetic risk score whether the individual is overweight or obese, it drives it up 10-fold, 20-fold, 30-fold. And this is true no matter what your genetic risk score is. So the lesson for this is that when your patient comes in and is overweight or obese and has a family history of diabetes, and they ask you, Doc, what can I do to prevent becoming type 2 diabetic? The answer is on this slide. Keep your body weight normal. Above all, keep your body weight normal, because obesity on top of the heredity of the diabetes genes is a huge, huge driver. And getting that body weight to as close to the normal range as possible is really going to reduce long lifetime risk of diabetes by the most. Another thing which I didn't appreciate but is now coming out, here's a recent article actually from The Lancet just a year or two ago, but in retrospect it seems to be obvious, is not only is there more obesity, but it's occurring at younger and younger ages. And I think those of you in the audience who are pediatricians probably appreciate this more than those of us who are adult physicians. This is obesity, prevalence of obesity by birth cohort. So it's by age and birth cohort. So if you go down here to these dark red lines, my father was born in 1909. So he's in this 1910 cohort. So when he grew to be 70 years of age and his cohort grew to be 70 years of age, only 10% of them would be obese, okay? I'm in the cohort that was born in the mid-1940s, so I'm in this orange cohort. And by the age of 70, my cohort has a 35% chance of being obese at the age of 70. My kids were born in the 1960s cohort, 1960s and early 70s cohort. So if you look at the 1960s and early 70s cohort, that's these yellow lines. My son's cohort has a 50% chance of obesity by the age of 50. And their kids' cohort has a 50% chance of obesity by the age of 30. So what's happening in society, and we can try to figure out what the drivers are, is not only is there more and more obesity, but it's occurring at younger and younger ages. And for those of you who see, young adults as well, you have to think about how this impacts our clinical management and our clinical treatment. And I would say the same is true in females. I showed you the males. The females show the same kind of progression, and it's even a little worse. So there's more female obesity than male obesity in young teenagers and young adults. Obesity definitely makes insulin resistance worse. This has been for many years. I would say that this is a complex relationship, and it varies from individuals. So this is a correlation between a measure of insulin resistance called SSPG, steady state plasma glucose. I won't explain it, except to just say the higher it is, the more insulin resistant you are. And here's a plot as a function of BMI. And you can see that there's a very strong linear correlation between obesity and insulin resistance. But at any given BMI, there are people like these individuals who can have low, which is normal SSPG, and people who can have very high SSPG, very insulin resistant. So not everybody reacts, and we all know that some individuals get very insulin resistant with very modest weight gains, and others can be very obese and seem to somehow be protected. And if we understood that, that's another important thing to think about. Part of this might relate to how the obesity actually involves inflammatory fat. So what happens is you gain adipose mass as fat cells get bigger, but also inflammatory cells, lymphocytes and macrophages invade the adipose tissue and make all kinds of inflammatory cytokines, change blood flow, etc. And so if we look at this, imagine this to be a muscle cell or a liver cell, and here's the fat cell. As the fat cell gets bigger, it's becoming more and more filled, the same fat tissue, with macrophages and other inflammatory cells. And these fat cells they're releasing free fatty acid that acts on the muscle liver to stimulate pathways involving kinases like JNK and mTOR and so forth. These serine phosphorylate IRS proteins and cause insulin resistance. The macrophages are stimulating inflammatory cytokines that also stimulate these pathways, leading to insulin resistance. They're secreting peptides and lipids that are also leading to this. So there are many things driving it. I would say that we don't know the relative contributions of each of these, but I would say it's not so simple. For example, even though there's a lot of inflammatory cytokines, immunosuppressive therapies do not necessarily alone relieve the insulin resistance, even though this contributes to it. So it's just one factor. And there are other things that are changing. Metabolites in the blood are also changing. But one thing I wanted to let you hear about this morning is another communication system that we're just appreciating a little bit now and is important in this process. And that is the ability of one cell to secrete microRNAs in little vesicles called exosomes. And fat's a great secretor of exosomal microRNAs. All tissues release them, but these exosomal microRNAs can also be taken up and create insulin resistance. And all of these things ultimately block insulin signaling and make the cell more insulin resistant. I want to spend just a minute on these exosomes because this is an area of current research that's very hot, not only in cancer biology and immunology, but in metabolism. Here's a fat cell releasing some of these little exosomes, and these exosomes contain proteins and lipids and little RNAs, microRNAs. And they're marked with a surface markers of protein called CD3 and CD9. And what happens is, to show you how important fat is, here's a profile of microRNAs in the serum of 4 normal individuals, human individuals, and red means high. And the two groups below are for individuals either with HIV-associated lipodystrophy or congenital generalized lipodystrophy. And look how all these high microRNAs become low microRNAs. So adipose tissue is secreting a lot of these microRNAs, and this is another way in which it communicates with muscle and liver and even brain to control metabolism. And so we did a study in conjunction with Sam Klein at Washington University, taking human subjects who underwent 15% or greater weight loss by diet or gastric bypass. And so they did this, and we looked at what happened to these exosomal microRNAs. And this is a profile of these exosomal microRNAs, and you can see in this volcano plat, a few microRNAs went up, but many, many microRNAs went down because fat's such a big source of microRNAs. And these microRNAs actually can target different parts. Here's that insulin signaling pathway, the receptor, the PI3 kinase, the AKT, and so forth. All these microRNAs tend to inhibit different parts of this pathway. So this change in circulating microRNAs may be another way that we affect insulin sensitivity, and it's a way for which yet we don't have any specific therapeutics. Finally, just a couple of last points, and then I'll allow a little time for questions. What comes out of fat is different for visceral fat and subcutaneous fat. I've already said visceral fat's the kind that causes insulin resistance. Subcutaneous fat's pretty neutral on insulin resistance, although it does affect metabolism. But I also want to remind everybody that there's a lot of interest now in metabolically active fat, primarily in the cervical and paraspinal region, called brown adipose tissue. This is adipose tissue which is filled with mitochondria, is involved in heat production, and can burn energy in addition to storing energy. And many years ago, we showed that this can be detected by PET scanning. Here's an individual PET scanned at thermoneutral conditions and then scanned after they've been exposed to cold, 61 degrees Fahrenheit, for about 2 hours. And you can see all of this increased PET activity, and much of this increased PET activity that's in supraglobicula and paraspinal regions, that's activation of this brown fat. And brown fat is good for you. White fat, not so good, but brown fat is good. Here's some data actually from your neighbors in New York who've taken something, I think I forget what the number is, but it's over 100,000 CT scans, or rather MRIs, and they've quantitated whether people have active brown fat or no active brown fat. And independent of BMI, if you have active brown fat, you have or let me say this the other way. If you have inactive brown fat, the brown bars are active brown fat. If you have inactive brown fat versus active brown fat, the active brown fat at every BMI have lower rates of type 2 diabetes. They have lower rates of dyslipidemia. They have lower rates of coronary artery disease, cardiovascular disease, congestive heart failure, and even hypertension. So brown fat is actually good for you at any body weight. And of course, white fat, particularly visceral fat, is bad for you. And we really still don't understand fully how this occurs. But part of this occurs by the fact that this brown fat is secreting, along with the white fat, is secreting these exosomal microRNAs. And some of those microRNAs are doing good. They're doing good at the liver, at the muscle. They're even getting into the brain and affecting activity at the brain. And I won't go any further with this because I want to leave a little questions. So I'm going to skip this slide. And I'm just going to say one final closing word about the microbiome. Superimposed on all this genetics and cell biology is the fact, of course, that when people are overeating and developing obesity, and they have the right genetics and the environment and the age, we know that this also affects the gut microbiome. And the gut microbiome is really interesting as a contributor to all of this, or as a modifier to all of this, because there are literally trillions of different microbes making literally trillions of different molecules. And these molecules that are coming out of the gut microbiome, depending on the pathway, create metabolites that are taken up into our portal veins and then circulate where they can affect liver metabolism and peripheral metabolism. And this little chart shows you just that some of the metabolites are short-chain fatty acids, amino acids like tryptophan, other metabolites, and bile acids. And the only thing I want to leave you with is that these metabolites can be good for you, or in some cases bad. And we don't know exactly which ones are which all the time. So we did a study that was just published a year ago in mice, where we compared metabolites in mice on a high-fat diet versus a chow diet. And what we found is that some metabolites coming out of the TCA cycle, called itaconate, mesoconate, and citriconate, if you took an insulin-resistant mouse, this is a mouse that's high-fat diet and it doesn't stimulate insulin action very well, you can restore that with certain metabolites, whereas other metabolites suppress it. So some metabolites are good, these are good metabolites, others are bad. These metabolites can also protect these liver cells from accumulating lipid. So here's lipid-rich hepatocytes from a high-fat fed animal compared to a chow. And if you treat these with itaconate, mesoconate, or citriconate, you reduce the lipid. So we can think of good guys and bad guys, and we have to learn which is which and then how to modify them. So I'm going to close here just to kind of bring all this together to say that what I hope I've shown you is that insulin resistance, in part driven by obesity, in part driven by genetics, is the fundamental driver of type 2 diabetes and metabolic syndrome. That there are genetic and epigenetic factors, but they're very complicated, and it isn't simply one or two genes. It's small contributions of literally hundreds of genes. So this is not something I think we can depend on yet to help us with our therapy. What we can depend on, I think, are two other things. We can depend on understanding the environment, the microbiome, and all of these pathways that they lead to. Inflammation, ER stress, mitochondrial dysfunction, oxidative stress, and so forth. All of these actually work on the insulin resistance. They all work on the beta cell dysfunction, and ultimately they all contribute to the pathogenesis of type 2 diabetes and metabolic syndrome. And with that, I'll just show a picture of my lab, some of their work I showed, and thank you very much for your attention, and again, the honor of giving this lecture.
Questions?
First, thank you, Dr. Kahn, for what I think was just a masterclass lecture, and just the information you presented it, how you presented it, the slide transitions. I would encourage all the trainees and young people in the room just to see how you did this presentation. So thank you so much.
Thank you.
My question to you, when early in my training, I had heard some talking about fat cells, which I don't know if some information has changed them, that they are immortal, that once you create a fat cell, it never dies, and that it was metabolically inert. Clearly, what you've shown is that they're absolutely not. So why is it that a fat cell is not inert and creating all this inflammation?
Yeah. Well, there are 2 points I'll make. First of all, there is something about what you said about fat cells being immortal. They're not obviously immortal, but so adult onset obesity is mostly hypertrophic. You have whatever cells you have, and they get bigger and fatter as you get bigger and fatter. Childhood obesity, and this was shown at the Rockefeller by Jules Hurst and many of his descendants many years ago, those individuals who have early onset or childhood obesity actually have both bigger fat cells and more fat cells. And you don't get rid of those so easily. So there is some truth to this notion that early onset obesity has this other dimension of hyperplasia as well as hypertrophy. And but all of these will respond, obviously, if you it's still energy balance prevails. And if you reduce energy intake to be less than energy output, everybody will lose fat, but it's probably true that those individuals who have childhood obesity will always have a greater propensity to refill those fat stores because they probably do have more cells. Cells don't live forever. These cells don't live forever, but they live for a long time, and that contributes a factor for sure.
Thank you. I agree. Magnificent lecture. You spoke at the beginning about exercise being exercise and diet being the only things we really have. But you didn't mention AMP kinase, which
Yes.
is improved with exercise.
Yes.
And that's been known for many years. And in Boston, you have Neil Ruderman, who advocated AMP for years. So what's become of that? Nothing has happened.
No, AMP kinase. So AMP kinase is another one of these protein kinases. It's present in many, many tissues. Its most prominent role initially was identified in muscle related to exercise. It's one of the kinases that's activated when we exercise, and it promotes energy metabolism by burning energy. It's also present in the brain, and it's involved in secretion of leptin and response to leptin. So it's definitely an important hormone. There so far are people have looked for sort of drugs to activate AMP kinase to get this exercise in a pill kind of thing. They haven't been very successful. And so I think it's an important thing, but we haven't been able to capture it for a therapeutic. It is part of the cascade, and it's probably, I would say, almost as important in the brain as it is in muscle. Because in the muscle, it's responding to exercise, but in the brain, it's actually playing a role in secretion of the hormones that control our appetite and food intake. So it's good.
Thank you so much. I also want to add my thanks for coming and for really a beautiful lecture. I'm going to play devil's advocate a little bit.
Sure.
We talked last night, and also you alluded to the fact that we don't always understand the mechanism of action of some of our really very effective drugs now. I mean, compared to 20 years ago, huge armamentarium of medications for diabetes,
Right.
finally effective drugs for treating obesity. Why would you say for this audience, is it important for us to continue to drive our understanding of the, the physiology and the sort of mechanisms there? Why can't we just figure out drugs that work and use them effectively?
No, that's a great question, Gene. A great question. I mean, certainly, even when we have drugs for which we know the mechanism of action, they often have effects beyond that mechanism. So what is our armamentarium? Well, we all know that the big thing to use now in obese individuals are GLP-1 analogs. Some of them are GLP-1 or multi-analogs for GLP-1, GIP, glucagon, and other things. And they're very effective in most people producing at least a significant amount of weight loss. This is probably not a physiologic effect of those hormones. Those hormones were really initially discovered because they promoted insulin secretion, but when given at pharmacologic dose, they promote weight loss. So I do think, and you could say the same thing is true about SGLT2 inhibitors. An SGLT2 inhibitor, we're allowing patients to get rid of excessive glucose load by actually peeing out the extra glucose, something that we also explored a long time ago. And the reason that they are important, and part of the reason they work, is that they are ignorant of the mechanism of the disease. They work on something that sort of like, having the kidney get rid of more glucose isn't really treating anything except hyperglycemia, really. It's reducing, and the osmotic effects of hyperglycemia, the metabolic effects. But it's an important therapy. So I don't think we should give up those therapeutics. Don't give up. Know those therapeutics, know how to use them for sure. I do think as physician scientist, what we would like in a perfect world, though, is to treat the primary driver. It's good to have antibiotics to treat an infection. It's better to prevent the infection if we could do it. And so I think that that's the way I look at this and what we need to do, because it may not be possible not just because of the cost, but just the practicality of people taking long-term GLP-1s. It's really both expensive to the system, it's demanding on the patient to be able to take a lifelong medication. And if we could really find the primary driver, I think that's always the best place to hit, if we can do it. Don't give up the other, but keep looking and keep thinking, whatever your disease interest is, about what are the primary drivers, because the closer we get to those, I think the more effective we'll be as physicians and physician scientists.
To add to that, you mentioned in the beginning that there's not really any drugs for diabetes that targets directly insulin signaling. Why is that? I mean, it's not for lack of trying, right?
Right. Well, I think that because this kinase cascade, this complicated cascade I showed you, is not so different from the cascade shared by other tyrosine kinases. And other tyrosine kinases include a number of oncogenes. So there's always been a worry that if you sensitize to insulin in a non-specific way, you will also maybe sensitize to epidermal growth factor and other things which can serve as oncogenes. I don't think we should give up on this. I do think a primary treatment, but I will say another challenge clinically is that we don't directly measure insulin resistance. You know, in contrast to glucose, everybody goes and gets a glucose in your patient. The closest thing you could come to insulin resistance would be to get a glucose-insulin ratio, but how many of you ever measure insulin in your patient? Probably almost 0%, maybe one guy here. Okay, and so we don't measure it, and that's only an indirect measure. If we had a blood test, you know, factor X in the blood was a great correlate, then we could, I think, have something that would stimulate not only looking at it, but then finding drugs to improve it. And that's another missing thing in our armamentarium, even after 100 years of insulin therapy.
Let me close with one question from the chat. Ayush Visaria asks, in someone on insulin therapy with severe insulin resistance, could the insulin therapy actually exacerbate downregulation, upregulation of insulin signaling pathways and make things worse?
Yes, so the question was, when you give a lot of insulin to somebody with insulin resistance, insulin actually downregulates its receptor. It causes post-receptor desensitization. And this is kind of a worry in theory. The real worry is not only that it downregulates the metabolic actions, but the growth actions, the ones going through the IGF receptor, may persist. And people have been worried about high and continuous insulin therapy in this regard because it might promote atherosclerosis, let's say, through endothelial and smooth muscle proliferation and so forth. So I do think it's a theoretical concern. I think the hyperglycemia uncontrolled is a bigger concern. So I think right now, in the absence of any other things to do, we really do need to try to control glucose. But it is, if we could do it without getting insulin levels very high, that would be optimal. And again, an insulin sensitizer would be the way to do it if we really had one.
Let me close by pointing out that Jesse Roth actually was a big proponent of the idea that hyperinsulinemia begets insulin resistance and maybe also a driver of obesity. So,
yeah.
A theme that he explored over decades. So again, I want to thank Ron Kahn for this terrific lecture. Thank you all for coming.
Thank you. I know it's a lot of basic science for a medical brain right Okay. Yeah, and we did it more or less on time, which I think was my other concern.
Wow. That was a
That was, wow. Other things, and I understand, and control of the flow. That is talk English. It was, I said it to Dr. Morosi before I said it to you, and I could follow the bread.
Yes, no, we need bread.
I'll take that as a compliment.
everything.
Oh, yes. But not, I was like, yes, but not as a science. I understand. I know Dr. Morrison explained it to me. It went right over my head. You can ask me. So that was a large. But the truth is, I can deal with the mechanics of the thing. And Bob did what he had to do. It was fabulous.
Now you lawyers have to see behind the
I'm working on it after all these years. Oh my God. Thank you so much. Yes, yes, and I know that Jesse sang your praises, Susan as well. I guess you guys did some socializing, a couple. Can I tell you a very funny story? That I love it. When I first came into Dr. Everett's class, I said to him, I wonder, you know, my cousin Jesse. I said, Jesse who? I said, Jesse Ross. And he said, he's a giant.